not dissolved in water. Okay. On the other hand, it can dissolve in organic solvents, which have non-polar properties. Okay. We are testing that property in very basic experiment number 1. Okay. In this experiment, you have to place 5 milliliters of distilled water and 5 milliliters of diethyl ether into 2 separate test tubes. Make sure that the tubes are clean and dry, and add 2 drops of oil into each tube. Make observations. Transfer the contents in the tubes containing diethyl ether and oil into a porcelain dish and allow to evaporate. Touch the remaining residue with your finger and recognize the oil is. So this is very basic experiment. So I'll show you the video. Then after that we can discuss more.
Today our practical is identification test for fat. We have a coconut oil sample, and the first test is the solubility test. For that, we are going to measure 5 milliliters of distilled water. And we are going to add it into the test tube, and then we are going to measure 5 milliliters from diethyl ether, and we have taken it into a beaker, and I am going to measure 5 milliliters of ether. Then I am going to add it into a separate test tube. Now I am going to take this coconut oil sample and add few drops of coconut oil into distilled water. And then I am going to take this ether test tube, and I am going to add few drops of coconut oil into the test tube containing ether. Now we will see the observation. In the sample with the diethyl ether, the coconut oil has been properly dissolved, okay? But here in the test tube we used with the distilled water, the coconut oil is not well dissolved. It you may see as a separate layer.
Okay. So I think there's no need to discuss about that further. That's a very basic test. Other than this test, solubility test, what are other tests, other very basic tests that we can use to identify fats or oils? There's one test in your advanced level chemistry syllabus. Very basic one. Can you remember that? Sudan test. Other than that, other than the Sudan test, yes, Sudan test again, a basic test to identify lipids. So other than Sudan test, yes, tel pallam pariksha. Then if you put fats or oil or whatever the compound that you need to test in a paper, then you will see oily spots. Okay, or tel tika කොලේට ගෑවට පස්සේ අර පේනවා නේද මොකද්ද අපි එකට කියන්නේ පාරභාෂක නෙමෙයි අර transparent වගේ oily spots පේනවනේ අන්න ඒක basic test එකක් විදිහට පාවිච්චි කරන්න පුළුවන් තෙල් පැල්ලම් පරීක්ෂාව කියලා අපි කියන්නේ so that one also you can be used to used as a qualitative test to identify lipids okay very basic one then I'll move on to the next test that is emulsification test okay so what is meant by an emulsion so since lipid is not dissolved in water but sometimes especially in food manufacturing we have to dissolve lipids in water you know that in most food processing steps we have to use lipids as well as aqueous substances okay so we have to mix these two and also most foods are emulsions we can say that there are natural emulsions as well as artificial emulsions in food products so what is the most common example for that food emulsions can you think even though lipid is not dissolved in water but naturally as well as in food processing we do mix in oil with water in that case we need to have this emulsification properties and we need to form emulsions can you give examples for that milk so milk contain fat as well as water okay so in milk can you see any I mean two layers okay if you keep milk for a longer period of time there you can see two layers clearly but in natural milk so there you cannot clearly identify two layers but you know in milk it contains lipids as well as water okay so it's a natural emulsion so other than that we can give many examples for such food items okay so emulsion is a mixture that used to mix two immiscible solvents immiscible means not mixing well so you know that water is not dissolved in oil is not dissolved in water so there if we need to have a mixture so we need to prepare a emulsion okay so in this experiment we're going to test the emulsification properties of lipids so for that we have to take three perfectly clean dry test tubes and 5 milliliters of distilled water 5 milliliters of salt solution and 5 milliliters of household detergent solution separately into test tubes add 1 milliliter of oil into each test tube and shake vigorously observe the changes over a period of 10 to 15 minutes okay so we will see the practical protocol and we will discuss about this deeply okay I think this is very interesting and very important timely important thing so we will discuss that later so please watch the video carefully
Now the next test is emulsification test. There also we are using the coconut oil sample, and we are using a detergent, a salt solution, and distilled water. Okay, we are going to measure 5 milliliters from each of these distilled water, salt solution, and the detergent. First, we are going to measure 5 milliliters from the distilled water. And we are going to add it into a test tube. Then we are going to measure 5 milliliters from the salt solution. Then we are adding it into another test tube. Next we are going to measure 5 milliliters from the detergent. And we are going to add it into a separate test tube. Then I am going to add 1 milliliters of coconut oil into each of these test tubes. And then 1 milliliter into the detergent. And finally, I am going to measure 1 milliliter and add it into the salt solution. Then I am going to shake them vigorously. And then we are going to let this sample for about 10 to 15 minutes, and we are going to take the observation. After 10 minutes, you may see the sample which we use salt and distilled water. There is a layer separation. That means they are not properly dissolved. But in the detergent, you may see it is well dissolved in the detergent.
Okay, then so as I mentioned earlier, as well as stated in the video, emulsification is the process of dispersing one liquid in a second immiscible liquid. Okay, and the resulting product is the emulsion. Okay, so as I explained earlier, milk is a good example for naturally forming emulsion. And other than that, butter is a very good example for the food processed emulsion. Okay, in emulsion there's two phases. One is you have learned that in your chemistry classes. I'm pretty sure about that in advanced level. One is that continuous phase and the dispersed phase. Okay, so milk is a oil in water type emulsion. It means the dispersed phase is oil, while the continuous or the large proportion is water. Okay. On the other hand, if we compare that with butter, butter is water in oil type emulsion. It means the larger portion is oil and the smaller portion is water. It means water is the dispersed phase and continuous phase is the lipid or the oil. Okay, so then in here we used a salt solution, a distilled water, and a household detergent solution. So when we use in a household detergent solution, at the end you observed a good emulsion. Okay, but on the other two solutions, especially in the distilled water, you observe two layers, oil layer and the water layer. So what is in the top? Is it oil or water? As well as in the solubility test, you observe that two layers. Yes, oil. What is the reason for that? Why oil goes to the top?
Density of the oil is relatively less.
No. Yes. As this is clearly answered, density of oil is usually less than water, so it goes to the top. And when we in this test, in household detergent solution, you observed emulsion. What could be the reason for that? Why it this household detergent formed a emulsion? Why other two cannot? Especially even though we didn't get that observation, in some salt solutions they can form emulsion, especially in our digestive system. It contains bile salts. So bile salts can form emulsions. So if you refresh your knowledge on digestion, food digestion, especially carbohydrate digestion and metabolism, so bile salts can form oil droplets, and it helps to digest that with lipases. So that term, I think you have learned that in your chemistry, thylodhikarane. Okay, then that is a similar process. Even we use sodium chloride like salt, it can form some kind of emulsion, but it's not much prominent. Okay, but okay, then why this household detergent solution formed a good emulsion? What could be the reason for that? Then what contained in the detergent solution, or maybe soap? Yes, sodium hydroxide, KOH. Okay, so let me explain that. So can you now see this presentation? Can you see the presentation?
Yes, sir.
Okay. So as I told, this is very interesting topic and timely important one. Okay. And let me explain why. So I know you have learned about soap production in your advanced level chemistry. So in salt production, we treat triacyl glycerides with caustic soda, that is sodium hydroxide, and the resulting product is soap and glycerol. Okay, so soap can be this kind of product. Depending on the type of the triglyceride we use, the resulting product can be sodium stearate, sodium palmitate, or any other product. So usually salt contains sodium stearate and sodium palmitate. Okay, so in this kind of sodium stearate or salt product contain, if you observe this clearly, a polar component as well as a non-polar component. So what is the polar component? So this sodium is acting as a head. So it's a polar component. As well as this hydrocarbon chain is non-polar component. And if we see the properties of these two components, this polar component is hydrophilic, while this non-polar tail is hydrophobic. Okay, hydrophilic and hydrophobic. Jalakami sahab jala patirodaka. So I don't know the exact Sinhala term for that. So in soap it contains a hydrophobic head as well as a hydrophilic head as well as a hydrophobic tail. Okay, so if you see the soap action, so as I mentioned, this soap or detergent contain sodium stearate or sodium palmitate. It means it contains a hydrophilic head as well as a hydrophobic tail. So when there is dirt or oily patches in your clothes, so this soap molecules can form micellars with them. Okay, you know that as I mentioned in the very first, there's a general principle in chemistry that similar properties dissolve each other. It means non-polar compound dissolve in non-polar compounds as well as polar compound dissolve in polar compounds. Then this when there is a dirt or oily patches, these soap molecules can form this kind of micellars. So here you can see if there is oily droplet, this hydrophobic or non-polar tail is attracted to this oily droplet and form this kind of structure. And if you observe this carefully, you can understand that this head is containing of sodium plus ions, and the charge of the head is plus. And when there's two such micellars, this micellar structures, what can you say? Will it be attracted or repulsed? මේ මිසෙල්ලා දෙකක් එක ළඟ තියෙනකොට ඒ දෙක attract වේද නැත්නම් ආකර්ෂණය වේද විකර්ෂණය වේද? Will it be attracted or repulsed? What can you say? There's a positive charge in the head. ධන ධන ආකර්ෂණ attract ආකර්ෂණ ධන charges attract වෙනවද නැත්නම් distract වෙනවද? ඒ කියන්නේ ආකර්ෂණය වෙනවද විකර්ෂණය වෙනවද?
විකර්ෂණය වෙනවා.
විකර්ෂණය වෙනවා. එතකොට මේ මිසෙල්ලස් දෙකක් එක ළඟ තියෙනකොට එක ළඟ තියෙන්නේ නැහැ විකර්ෂණය වෙනවා. එතකොට it can repulse and disperse in the solution. So that's the soap action. That's how soap can remove patches in your or remove dirt in your clothes. Okay, පැහැදිලිද? අපේ කුණු පැල්ලම් ගොඩක් ඒවා oily patches. එතකොට මේ oily patches වටා මේ soap molecules වල මේ වගේ micellars හදන්න පුළුවන්. එතකොට මේ micellars කිහිපයක් මිසෙල්ලාස් හදනකොට මේ මිසෙල්ලාස් කොයිලාකවත් එක ළඟ ඇට්‍රැක්ට් වෙන්නේ මේ ධන ආකර්ෂණ තමයි පිටත තියෙන්නේ. එතකොට මේ ධන ආකර්ෂණ තව මිසෙල්ලාට ධන ආකර්ෂණයක් තියෙනකොට මේ දෙක රිපල්ස් වෙනවා. එතකොට ඒ කියන්නේ වතුරේ ඩිස්පර්ස් වෙලා යනවා. එතකොට අපි රෙදි උදනකොට ඔයගොල්ලෝ දන්නවා එකම වතුර නෙමෙයි වතුර රීසයිකල් නැත්නම් මේ වතුර කීපවතාවක් පාවිච්චි කරනවා. එතකොට මේ වතුරත් එක්ක කුණු පැල්ලම් යනවා. So that's the soap action. Then why we tell always in the television and whatever the media you are using nowadays, then you are hearing that you have to wash your hands using soap to avoid the COVID-19 infection. So what can these soap molecules do to COVID-19, this virus? So that is similar to this action. That's why I'm talking this. Okay, it is not that will not I mean there's no point of talking the theoretical or practical things that has no applications. Okay, we have to apply all these things to our day-to-day life. Okay, otherwise there's no use of our knowledge. Okay, then how what is the reason for telling that?
protein ආවරණය කාටද බ්‍රේක් කරනවා?
ඔව් ගොඩක් හොඳයි. අපේ මේ COVID-19 virus එකේ තියෙන්නේ protein ආවරණයක් එක්ක lipid ආවරණයක් එක්ක protein spikes. මේ spike protein තියෙනවා, මේ ආවරණයේ තියෙන්නේ lipids. එතකොට මේ soap molecules වලට hydrophilic head එකක් තියෙනවා, hydrophobic නැත්නම් non-polar tail එකක් තියෙනවා. එතකොට මේ non-polar tail එකට මේ lipid ආවරණය විනාශ කරන්න පුළුවන්. ඒ වගේම විනාශ කරන්න බැරි වුණත් අර micellar action එකකින් හින්දා මේක අයින් කරන්න පුළුවන්. අපේ අතේ මේක you know that viruses are very small things and they can so even within a very small space there can be millions of viruses. Okay, so even though these soap molecules cannot kill or cannot damage this lipid membrane, it can form micellars and remove that with water. So that's why we advise, health professionals advise to wash hands with soap, whatever the soap. It should not be always Lifebuoy or that Dettol or that so-called disinfectant soap. Even sunlight can do the same thing. Okay, so that's the reason. So here you can see soap molecules can form micellars that is similar to that we discussed in the washing action. So here's the skin and this dot dirt substances can have virus. So when removing the dirt substances, so virus can remove from our skin, even though this can form micellars with the single virus cells. Okay, since virus is very small and these molecules are very large, the formation of micellars sometimes we cannot observe that clearly, but the action is similar to that of cleaning or washing clothes. Okay, did you understand that? So emulsion එකක් හදනවා කියලා කියන්නේ මේ වගේ micellars යන්න පුළුවන් continuous space එකට. ඒ කියන්නේ water තිබ්බොත් එහෙම water එකේ හොඳට පැතිරෙන්න පුළුවන්. පැහැදිලිද? අපි දැන් වතුරට තෙල් ටිකක් දැම්මොත් එහෙම තෙල් උඩ පාවෙනවා, වතුර යට තියෙනවා. එතකොට මේ දෙක අතර emulsion එකක් නැත්නම් mix එකක් හැදෙන්නේ නැහැ. interface එකක් තියෙනවා, උඩ තෙල් තියෙනවා, යට තියෙනවා. emulsion වතුර. එතකොට මේ වගේ emulsifying agent එකක් නැත්නම් soap වගේ දෙයක් දැම්මොත් එහෙම වෙන්නේ මේ වගේ මිසෙල්ලාස් හදලා තෙල් වතුරේ disperse වෙන්න පුළුවන්. So that helps to form an emulsion. So in this case, soap can act as a surfactant. Surfactant means a molecule or compound which can interact with polar phase as well as non-polar phase. So that's quite obvious because this soap contain hydrophilic component as well as the hydrophobic component. Okay, so it can act as a surfactant. Okay, so that's the action of soap in forming emulsions as well as protecting against COVID-19. Okay, so is that clear for you?
Yes, sir.
මේක රිලේට් කරේ අපි මේ අපිට application නැති science එක ඉගෙනගෙන කිසි වැඩක් නැහැ. But biochemistry we can relate that to our day-to-day life and it is very important subject. तर මේ action එක තමයි emulsion එකක් හැදෙද්දිත් වෙන්නේ. පැහැදිලිද?
excuse me sir.
ඔව්.
so ඒතර ඔය service station වල garage වල එහෙම සර් මේ water purify system එක අර chemical එකක් දානවා කියලා කරන්නේ තෝකම සර්?
ඔව් මේ වගේම දෙයක් තමයි. මේ වගේම දෙයක් තමයි කරන්නේ. තොර grease අයින් කරන්න අපි high pressure එකකින් physical action එකක් තියෙනවා. මොකද high pressure use කරලා පස්සේ high pressure water use කරලා පස්සේ මොකක් හරි දෙයක් තියෙනවා නම් ඒක force එකත් එක්ක අයින් වෙන්න පුළුවන්. අපි ඒක facilitate කරන්න දාන්නේ මෙන්න මේ වගේ detergent එකක් තමයි. Okay, so cleaning and sanitizing is two different things. So basically in food science we talk about that. In cleaning we intend to remove the physical dirt materials. In sanitizing we intend to lower the microbial load. Okay, so when we considering about COVID-19 protection, we have to sanitize, not clean. අපි පාවිච්චි කරන්නේ පිරිසිදු කරනවටත් වඩා සනීපාරක් නැත්නම් sanitize කරන්න. අපි කියන්නේ පිරිසිදු කරලා තියාගන්න කියනකට වඩා අපි කියන්නේ සනීප කියන්නේ sanitize කරන්න කියලා අත් දෙක. ඒ කියන්නේ මේ detergent එකක් දාලා වතුරෙන් විතරක් ඒක වතුරෙන් ක්ලීන් කරන්න පුළුවන් නම් වතුරෙන් sanitize කරන්න බෑ. So sanitizing action එක වෙන්නේ මෙන්න මේ දේ. Okay, so that is related to our experiment. That's why I explained that to you. Okay, so that this thing happened in our digestive system also. So bile salt or pitta amla, so they can do this so-called chylon formation or thylodikaranaya. So it means the oily parts in our food can break into small droplets, and so a lipase enzyme can effectively act on these droplets. Okay, baby soap වලින් COVID-19 virus එක විනාශ කරන්න පුළුවන් possibility එක අඩුයිද? අපි හරියට බැලුවොත් එහෙම දෙයක් අපිට හිතන්න පුළුවන්. නමුත් ඔය ලයිෆ් බෝයි වලයි සන්ලයිට් වලයි ලොකු වෙනසක් නැහැ. ඔකේ අපි කියනවා මේ TF value එක කියලා. අර ඒ values අපි කියන්නේ toilet soaps කියලා කියන්නේ. toilet soap වල ඊට පස්සේ ඔය cleaning soaps වල වෙනස්. ඒක තමයි ඔය baby soap වලයි මේ නැත්තම් ලයිෆ් මේ clean කරන්න පාවිච්චි කරන ඒවා වෙනස. නමුත් මේ action එක හැම එකටම පොදුයි. baby soap උනත් ඒක sunlight උනත් life boy උනත් නැත්නම් pears උනත් baby cherry උනත් එකම action එක තමයි වෙන්නේ. මොකද හැම එකේම තියෙන්නේ sodium stearate. මේකේ concentration එක ඔය intended use එක අනුව වෙනස් වෙනවා. Especially in baby soap production we have to be very careful. And so we are using that for a vulnerable group. So the chemical residues in that soap is very less, and we have to produce that in restricted, I mean very careful processing conditions. So if there's chemical residues and higher concentrations in concentrations of hazardous material, it can cause a lot of harm. Okay, so I cannot directly say that baby soap have less action against COVID-19. Yes, the WHO recommend wash your hands whatever the soap you have. Okay, but in baby soaps it contain less TMF value. Sanitizer හා soap වලින් ගොඩක්ම හොඳ මොකක්ද COVID-19 වලට? Sanitizer හා soap වලින් ගොඩක්ම හොඳ alcohol sanitizer වල action එක මීට වඩා වෙනස්. දැන් අපි ඔය alcohol වලින් directly it can kill the virus. So it is better to use sanitizer if you have. But you know that when we use the sanitizer for a longer period of time, it can cause some allergic reactions and other things. And cost-wise, sanitizer is more expensive than soap. So you have to scrutinize. But the action of alcohol sanitizer and soap is different. Alcohol sanitizer ke soap polai action එක වෙනස්. Alcohol can kill microbial cells, virus, bacteria, and other microbial cells. So it does not have this kind of emulsion formation and formation of micellars and removing the microbial cells. Okay, it can directly kill the microbial cells, not forming emulsions and removing from our surface of skin. Okay, but soap has that action that I explained earlier. Okay, so මේකත් පුංචි ටෙස්ට් එකක් emulsification test එක කියලා. මොකද මේ lipid වලට මේ detergent නැත්නම් bile salts වගේ දේවල් එක්ක emulsions හදන්න පුළුවන්. But you cannot see this property in carbohydrates. You know that. So if you use monosaccharide, it dissolves well in water. Okay, and even starch cannot form emulsions. It is not considered an emulsion. It can partially dissolve in hot water. You know, the dissolving in hot water, the action is different than this. It is not forming micellars. The granular form is damaged, and the amylose and amylopectin get separated. And you know that the dissolving power of amylose and amylopectin is different. And as a result of that, starch can dissolve in hot water. Okay, you have to understand the action. Okay, so I'll move on to the next test. Okay, other than that, you have learned about one test to identify the presence of unsaturation, or to distinguish saturated and unsaturated fats or fatty acids. That's a very famous test that you have used in your organic conversions. What is that test? Can you remember? ඊට අමතරව ඔයගොල්ලෝ ඕගනික් පරිවර්තන වගේ දේවල් වලදී ඊළඟට ඉගෙන ගත්තා ගොඩාක් famous test එකක් unsaturated bonds අඳුර ගන්න. මොකක්ද?
BR2 Edgar.
Yes, very good. Yes, if we add bromine water to fat or whatever the compound we need to test on saturation and unsaturation. So if there is a double bonds, this BR2 will lose its color. You know, BR2 has a brownish color, and when there is presence of double bonds, BR2 can attract to that double bonds and form BR intermediates, and as a result of that the color will turn into colorless. Yes, adding BR2 bromine test. Okay, other than that there are two major tests. I don't think you have learned that in your chemistry, advanced level chemistry. Lieberman Burchard test, as well as Salkowski test to test cholesterol, particularly to test the presence of cholesterol. Okay, Lieberman, if you are interested, search about those two tests. We unfortunately we don't have that two tests in our syllabus. Okay, if we have that, it is good, and I have suggested that for the next curriculum. Lieberman Burchard test and Salkowski test. Using those two qualitative tests, we can identify the presence of cholesterol. You know, we have a very concern about cholesterol. So, so it can cause lots of health consequences. I mean, adverse health conditions in human. Okay, so it's a kind of lipid. Okay, the properties of cholesterol is somewhat different than the true fats and lipids. Okay, Lieberman-Burchard test and Salkowski test. Okay, so as the next practical, we have to discuss about quantitative analysis of fats and oil. So in this experiment, we are going to determine acid value of a fat or oil sample. Okay, so acid value. So there's a definition for acid value. We have to understand that first of all. Okay, acid value is defined as the number of milligrams of potassium hydroxide required to neutralize the free acids in 1 gram of oil. Free acids are formed when the oil are oxidized into fatty acids and glycerol. This gives an idea about the level of rancidity of the given oil. So what is meant by rancidity or rancidification? Is rancidification or rancidity. So what will happen if oil or fat get rancidified? What are the resulting products? Rancidification. What is the single term for rancidification? Muduim, yes, muduim. That's not Futibawni. That's putrefaction. That's for proteins. Muduim. Then if I talk about rancidification within 1 minute. So what are the causes for rancidification? There can be various causes. මුඩුවීමට හේතු වෙන්න පුළුවන් මොනවද?
Microbial activity.
Yes, microbial activity. So you know that lipases like enzymes can formed by microorganisms, and these enzymes can rancidify fats and oils and degrade them into glycerol and 3 fatty acids. If you consider glyceride, triglyceride, it can decompose into fatty acids and glycerol. So other than microorganisms, what are the causes? Muduwimata hetu wunna. You know these things.
Oxygen.
Yes, oxygen. Oxidative rancidity, exposing to air. Yes, as near Nimtara or Nimtara, and you answered yes, oxygen, oxidative rancidity. And other than that, water also can cause to rancidification. That is called hydrolytic rancidity. Okay, so when oils get rancidified, that will form free fatty acids. Okay, then level of free fatty acids presence in oil or fat is used as a quality assurance measure or quality measure of that oil. What can you say? If there is a high level of free fatty acids, quality of that oil is high or low. What can you say about that? Product free fatty acids තියෙනවා නම් ඒ oil එකේ quality එක වැඩිද අඩුයිද? So usually in food industry this is used as a quality measure. Quality එක අඩුයි. Free fatty acids තියෙනවා කියලා කියන්නේ ඒ oil එක නැත්තම් fat එක decompose වෙලා නැත්තම් rancify වෙලා කියලා. Rancify එක මුඩු වෙනවා කියන එක හොඳ දෙයක්ද නරක දෙයක්ද? පොල් තෙල් ඔයගොල්ලෝ කඩෙන් ගන්නකොට මුඩු වෙච්ච පොල් තෙල්ද ගන්නේ? නැහැ. මුඩු වෙනවා කියලා කියන්නේ ඒක that can form other chemical products as well as unpleasant flavor as well as odors. Yes.
Excuse me, free acid වෙනවා කියන්නේ මොකද්ද? free acid වෙනවා කියන්නේ bond එකකින් වෙන් වෙනවද?
Bond එකකින් වෙන් වෙනවා. So let me explain that. Yes, lipid rancidification. So this is triglycerides, that is the natural form of fats or oils. So when there's favorable conditions like water, oxygen, microbial enzymes, so these triglycerides can decompose into glycerol and fatty acids. So you use if one triglyceride decomposed, it can form one glycerol molecule as well as 3 fatty acids. So decompose වෙනවා කියලා කියන්නේ fatty acid content එක වැඩි කියන එක. So fatty acid content එක වැඩි කියලා කියන්නේ free fatty acid වැඩි කියලා කියන්නේ. So there's a problem. Okay, so there can be a problem. Okay, this some fatty acids can form unpleasant flavors as well as unpleasant odors. That's why some decomposed oil is more is having unpleasant flavors as well as odors. Okay, so when there's a higher fatty acid level, that means the quality of that oil is less, as well as this can be used as a measure of the shelf life of a oil. Okay, when oil is stored for a longer period of time, usually fatty acid content is higher. That means that oil is old. Okay, පැහැදිලිද? ඒක තේරුම් ගන්න ඕන මුලින්ම. Is that clear for you? Tulakshi, is that clear for you? Okay. So I will show you the video. After that we can discuss this about this more.
Today our practical is determination of acid value. For that, we are using a test sample of coconut oil. I have already measured 5 grams of coconut oil into this conical flask, and then I'm going to measure 12.5 milliliters from each of ethyl alcohol and ether. Now I'm going to measure 12.5 milliliters of ether, and I'm going to add it into the conical flask. Then I'm going to measure 12.5 milliliters of ethyl alcohol. Then again I'm going to add it into the conical flask. Then I'm going to add few drops of phenolphthalein indicator. The next step, we are going to keep this in a water bath. For this experiment, we are also going to prepare a blank solution. That means without the coconut oil, we are going to add all the other chemicals and do the similar procedure as we did in the coconut oil sample. I have already measured 12.5 milliliters of ethyl alcohol. Now I'm going to add it into this conical flask. Then I have measured 12.5 milliliters of ether, and we are going to add it into the same conical flask. Also, I am going to add few drops from the phenolphthalein. And now we have to keep the sample in the water bath for the proper, we have to dissolve it properly, therefore we are placing it in the water bath. We are going to place our conical flask in this water bath until the mixture gets properly dissolved. Now you may see the mixture is properly dissolved, and we are going to take this out of the water bath and we are going to titrate it. Next, we have taken the sample out of the water bath and we are going to titrate it against the 0.1 KOH. Okay. For that, I have already washed the burette properly in the correct way, and I have filled it with the KOH, and I'm going to titrate it with the sample. Okay. Now let's start the titration. Now we are going to titrate this hot sample, and the end point of this is the pink color should persist for a few minutes. Keep in mind, do not shake this vigorously. the endpoint of this test is the point where the pink color persists for a few minutes, and after you get the endpoint, you have to note down the reading. Okay. Then we are going to practice the same procedure for the blank solution, and we are going to titrate it against the KOH. Here also you may see we get the persisting pink color, and now we have to record the reading of this burette. Okay.
Okay, so in this experiment, to determine acid value, you have to weigh about 5 to 6 grams of coconut oil. You have to measure record the exact weight that you are taking into a clean diachronical glass, and you have to add 12.5 milliliters of each ethyl alcohol and ether, and few drops of phenolphthalein, and you have to heat the mixture on a boiling water bath until all the oils has dissolved well. Titrate the hot solution with the standard 0.1 in potassium hydroxide. If oil drops are floating heat for a short time, the faint pink color should persist for about a minute at the end point. Carry out the experiment in duplicate and do the blank using the reagent. Calculate the acid value of the coconut oil. So this is a acid-based titration at the end, but there are importance of each step. Okay. In here we are adding ethyl alcohol as well as ether. So what can you say about the polar or non-polar properties of ethyl alcohol and ether? Ethyl alcohol, is it a polar solvent or non-polar solvent? So what do you think? Ethyl alcohol, ether, we might think that oil is non-polar and it is well dissolved in ether, and then why we use ethyl alcohol? So ethyl alcohol is a polar solvent. You know that ethyl alcohol means ethanol. Okay, so when there are decomposed products, fatty acids can be also there. Okay, then if you see the structure of fatty acids, fatty acid contains COOH content at the end. Okay, so so you know that in fatty acids, based on the carbon number, 6 to 26 carbon can be there. Okay, so some fatty acids, especially that terminal COOH, is well dissolved with polar solvents. So in order to dissolve the content fully, we use ethyl alcohol as well as ether. Okay, මේ fatty acids decompose වුණාට පස්සේ oil එකේ සාමාන්‍යයෙන් තියෙන්නේ naturally triglycerides. නමුත් rancidify වුණාට පස්සේ free fatty acids තියෙන්න පුළුවන්. Free fatty acids වල COOH end එකක් තියෙනවා. කාණ්ඩයක් තියෙනවා. එතකොට මේ COOH, you know that it is a kind of polar functional groups. Then we use ethyl alcohol to dissolve that polar functional group having fatty acids well in that solution. So in here we use a polar solvent as well as non-polar solvents, and in order to increase the rate of reaction, we do heat this for a short period of time using the water bath. Then What do you think? Can we use an open flame to heat the content? What do you think? අපිට open flame එක පාවිච්චි කරන්න පුළුවන්ද මේ content එක රත් කරන්න? Yes, you know that organic solvents can easily catch fire. So we never use open flame when we use in organic solvents like ether as well as ethyl alcohol can catch fire. So if you have a sanitizer with you, spray it on your table and light with fire, then you will observe a fire. Okay, ඔයගොල්ලන්ගේ sanitizer බෝතේ පොඩ්ඩක් මේසෙ උඩ ස්ප්‍රේ කරලා ගිනි කුරක් ගහලා බලන්න හොඳට ඇවිලෙනවා. You know that organic solvents can easily catch fire. Okay, yes, okay. Then after that you have to boil that to mix that well. And if we not provide heat here, there will be two layers. Okay, since we use polar as well as non-polar solvents, you know that they are not dissolve each other, but those two are organic solvents. So they dissolve in somewhat to increase the dissolution or dissolving each other. We provide heat here. Okay, and after that, after dissolving these two, so when two layers are there, we cannot observe the end point clearly. So that's another point, important point of boiling the content before do the titration. Okay, soon after getting that from the water bath, boiling water bath, especially from the boiling water bath, you have to do the titration against 0.1 in potassium hydroxide. Okay, then in the end point, you must have obtained persistent pink color for at least about 1 minute. And if you heard what our demonstrator explained to you, you should never do the vigorous shaking. So if you do the vigorous shaking in this titration, atmospheric carbon dioxide can dissolve here and form carbonic acid. So when carbonic acid formed, some amount of potassium hydroxide can consume for that. Therefore, we never do vigorous shake. අපි හයියෙන් මේක දිය conical flask එක හයියෙන් shake කරොත් එහෙම atmosphere එකේ තියෙන carbon dioxide dissolve වෙන්න පුළුවන්. එතකොට carbonic acid දෙන්න පුළුවන්. Carbonic acid neutralize කරනත් potassium hydroxide consume when hinda. So the end point will be higher than the true value. So we never do vigorous shake. So that's one important point in this titration. Okay, so in this titration, you have to do a blank titration also. So what is meant by a blank titration is that we without using the sample or the main matrix, we do the titration for other regions. The reason for doing a blank titration is that so ethyl alcohol as well as ether can contain some level of acidity. Okay, since we use ethyl alcohol and ether as the reagents, so some amount of potassium hydroxide can consume to neutralize the acidity of ethyl alcohol and ether. Okay, so to calculate the acidity or the volume consumed to neutralize that reagents, we do a blank titration. Okay, blank titration එක කියලා කියන්නේ oil නැතුව අපි පාවිච්චි කරන හැම reagent එකක්ම දාලා titrate කරනවා. මොකද අපි පාවිච්චි කරන reagents වල ethyl alcohol and ether are slightly acidic, so some amount of potassium hydroxide can consume to neutralize that acidity in the main titration. Okay, to overcome that issue, we do a blank titration. Okay, so that are the reasons for each step in this experiment. And also this acid value is in some books I have seen that this is referred as acid number, neutralization as acid number or neutralization number. Okay, acid value කියන එක neutralization number කියලා සහ acid number කියලා සමහර තැන්වල in some biochemistry books I have seen that. Okay, so there's a specific definition for acid value. The number of milligrams of potassium hydroxide required to neutralize the free acids in 1 gram of oil. Okay, so that's the definition of acid value. So you have to keep that in mind always. Okay, so if I ask that, if I explain that again, this procedure, ඔයගලන්ට මුලින්ම ග්‍රෑම් 5ක් 6ක් අතර ප්‍රමාණයක් coconut oil ගන්න ඕනේ. මේ ගන්න ප්‍රමාණය අනිවාර්යයෙන්ම රෙකෝඩ් කරගන්න ඕනේ, because in the calculation you have to use the weight. Okay, ගත්තට පස්සේ conical glass එකට ගන්න අපි titrate කරන්න ලෑස්ති කරගෙනම ග්‍රෑම් මිලිලීටර් 0.5ක් ethyl alcohol සහ ether add කරන්න ඕනේ. පිනොප්තේන් මෙතන පාවිච්චි කරන ඉන්ඩිකේටර් එක විදියට ඒකත් add කරනවා. ඊට පස්සේ මේ content එක conical glass එකේ තියෙන content එක water bath එකක තියලා පොඩ රත් කරනවා. මොකද මෙතන ethyl alcohol සහ ether තියෙන නිසා මේ properties වෙනස්. ether කියලා කියන්නේ non polar solvent එකක්. ethyl alcohol කියලා කියන්නේ polar solvent එකක්. එතකොට මේ දෙක ඔයගොල්ලෝ දන්නවා හොඳට දිය වෙන්නේ නැහැ. නමුත් මේ දෙකම organic solvents. organic solvents වල ටිකක් දිය වෙනවා. ටිකක් නෙමෙයි හොඳට දිය වෙනවා. නමුත් අර polarity එකේ ප්‍රශ්නේ හින්දා දිය වෙන එක අපිට 100% ensure කරන්න බෑ. එතකොට මේකේ layers දෙකක් තිබ්බොත් එහෙම titration එකේදී අපිට color change එක හරියට බලා ගන්න බෑ. ඒකින්ද තමයි අපි රත් කරන්නේ. රත් කරනකොට අපිට මේ layers දෙක නැති වෙනවා. ඒ කියන්නේ හොඳට දිය වෙනවා වගේ පේනවා. එතකොට ඊට පස්සේ අපි මේක boiling water bath එකෙන් ගත්තට පස්සේ ගොඩක් වෙලා තියන්න නැතුව අපි sodium hydroxide එක burette එකට අරගෙන ඒකේ titration එක කරනවා. එතකොට end point එක වෙන්නේ persistent pink color එක. Okay, persistent pink color. ඔගොල්ලෝ titration එක කරද්දී titration මේ burette එකෙන් බිඳුව වැටෙන තැන pink color වෙන්න පුළුවන්. නමුත් ඒක නෙමෙයි end point එක. ඔයගොල්ලෝ මුළු solution එකම pink color වෙන්න ඕනේ. ඒක අඩුම ගානේ විනාඩියක්වත් තියෙන්න ඕනේ. That's what meant by persistent pink color. Okay, or constant pink color. So in this method we have to do a blank titration. Blank titration එකක් කරන්න ඕනේ. මොකද මේ ethyl alcohol සහ ether කියන ඒවත් slightly acidic. ඒකේ ප්‍රශ්නේ තමයි අපි පාවිච්චි කරන potassium hydroxide මෙන්න මේ වගේ acidity වලට acidity එක neutralize කරන්නත් බය වෙන්න පුළුවන්. ඒ කියන්නේ ethyl alcohol සහ ether වල. එතකොට අපි ගන්න reading එක burette reading එක වැරදි. ඒක overcome කරන්න නැත්නම් ඒක correct කරන්න තමයි අපි blank titration එක කරන්නේ. Okay, so that is the simple explanation of this procedure. Okay, so do you have any questions regarding this? So we have to do a calculation. So I request you to take your calculators. Please understand this properly, because this can also appear in your exams. Okay, ඔයගොල්ලෝ ගාව calculators දෙනවා නම් ළඟට ගන්න. හරි, calculation එකට යන්න කලින් මේ procedure එකේ මොකක් හරි ප්‍රශ්නයක් තියෙනවද? අයි අපි මේ දේවල් කරේ කියන්නේ in your discussion you have to write about the importance of each step and why we do this.
Excuse me sir.
Okay, can
Excuse me sir. Sir blank test එක සර් මුලින්මද කරන්නේ නැත්නම් මේ අන්තිමටද කරන්නේ?
ඒක ප්‍රශ්නයක් නෑ. මුලින්ම කරත් කමක් නෑ, අන්තිමට කරත් ප්‍රශ්නයක් නෑ. සාමාන්‍යයෙන් අපි කරන්නේ මුලින්ම blank titration.
තව පාරක් explain කරන්න පුළුවන්.
හරි. තව පාරක් explain කරන්න. Okay. So this is to determine the acid value. It means the available free fatty acids in a given sample. So there's the definition for acid value is that the number of milligrams number of milligrams of potassium hydroxide, not grams, milligrams of potassium hydroxide required to neutralize the free acids in 1 gram of oil, specifically 1 gram of oil. Okay. So in order to calculate acid value, we have to take roughly about 5 to 6 gram of coconut oil, and you have to record the value into a conical flask. And after that, you have to add 12.5 milliliters of each ethyl alcohol, that means alcohol and ether, with few drops of phenolphthalein. Phenolphthalein we use as the indicator here. Okay. After that, after getting phenolphthalein, ethyl ether, ethyl alcohol and ether, along with the coconut oil sample that we measured, we have to boil that on a water bath for about 1 minute. Okay, until this content dissolved well. After that, you have to get that from the water bath and immediately do the titration against 0.1 in potassium hydroxide. It means KOH. Okay, so at the end point, you will observe a persistent pink color or constant pink color. Okay, so in order to increase the accuracy of this experiment or this titration, you have to do a blank titration, and you have to at least do this titration for another 3 times. Okay, so by getting when we know the exact volume, exact weight we get, and the final burette reading, we can calculate the acid value. Okay, so when we calculate in the acid value, you will see. We'll see how this works. Okay. Acid transedification කියලා කියන්නේ මෙන්න මේක triglycerides, glycerols of fatty acids වලට කැඩෙන එක. එතකොට naturally fats or oils exist as triglycerides or true lipids, and when favorable conditions like H2O, oxygen, or some microorganisms are there, so it can decompose into glycerol and fatty acids. That is called rancidification. Okay, that is the chemical decomposition of lipids. Then, yes, the titration, what is happening is this. We use potassium hydroxide in the burette, and when there are free fatty acids in the sample we took, it can react with potassium hydroxide and form this product and water. Okay. So what can you say about, is this a strong acid, strong base titration, or strong acid, weak base titration, or strong base, weak acid titration? What kind of titration is that? Make a dubala amla prabala bashma titration nekadda, prabala amla prabala bashma titration nekadda, prabala amla dubala bashma titration nekadda? What do you know about this titration by looking at this? What can you say? Very good. This is a kind of a weak acid and strong base kind of titration. Okay, so in here you know that for weak acid, strong base like titrations, we can use phenolphthalein as the indicator. So that's why we used phenolphthalein. Okay, so in here, so I use this calculation to explain this to you. So please record these things. So I have done four titrations. For the first titration, I took 5.000 gram. Then for the second titration, 5.0010 gram. So if you are given to measure exactly 5 grams, so sometimes you may not measure that 5 grams accurately. It's okay to use. It's okay to get 5.0010 or 5.0005, and you have to record that value and use for the calculation. okay අපිට 5යි 6 අතර ග්‍රෑම් ගන්න කිව්වට පස්සේ 5ක් ගන්න පුළුවන් 6ක් ගන්න පුළුවන් ඒ අතර ඕන ප්‍රමාණයක් ගන්න පුළුවන් නමුත් අපි මේ weight එක record කර ගන්න ඕනේ මොකද මේ weight එක අපිට වැදගත් වෙනවා calculation so in here I have done 4 titrations 5.00 gram for the first titration 5.0010 gram for the second titration 5.005 grams for the third titration and 5.000 gram for the fourth titrations. Okay. And the molecular weight of KOH is 56 gram per mole. So have you recorded this? So do the calculation with me. So these are the blank titration readings. So these are the KOH volume consumed to neutralize reagents we use. It means ethyl alcohol and ether. First reading 0.05, second reading 0.05, and third reading also 0.05. Same reading obtained. So these are the blank titration readings. And these are the titration readings. For the first titration, it consumed about 6.35 milliliters. Second reading, we got as 10.25 milliliters. And as the third titration, we got about 10.25 milliliters. As the fourth reading, we got about 10.10 milliliters. Okay. Then මෙතනදී මට කියන්න අපි titration reading එක මෙතන අපිට average value එකක් ගන්න පුළුවන්ද ගන්නවා නම් මොන readings ද consider කරන්න ඕන මේක හරියට තේරුම් ගන්න මම දන්නවා ඔයගොල්ලෝ ගොඩක් කට්ටිය සමහරවිට මේක වැරදියට තේරුම් ගන්න පුළුවන්
sir මේ titration reading එකෙන් sir blank titration reading එක අපි අඩු කරන්න ඕනේද
ඔව් අඩු කරන්න ඕනේ මෙතන අපි titration reading ගේ average එකක් ගන්න පුළුවන්ද average එකක් ගන්නවා නම් මොන readings ද අපි ගන්න Actually, this kind of titrations, we cannot get the average. That's the proper practice. Okay, you have to think. You have to understand that. අපි මේ වගේ titrations කරද්දි මම දන්නවා ඔයගොල්ලෝ ගොඩක් තැන්වලදී readings average එක ගන්න මේ වගේ තැන්වලදී average එක ගන්න බෑ කොයිලාකවත්. ඒක වැරදි. හේතුව තමයි අපි ගත්ත weights වෙනස්. අපි ගත්තේ මොකද අපිට ග්‍රෑම් 5ක් කියලා මෙෂර් කරන්න කියලා ගියොත් අපිට හරියටම කොයිලාක ග්‍රෑම් 5ක් මෙෂර් කරන්න බෑ. ඒක ග්‍රෑම් 5යි 5 බිංදුවක් වෙන්න පුළුවන්. 0.1යි බිංදුවක් වෙන්න පුළුවන් 5යි 5 5ක් වෙන්න පුළුවන්. එතකොට අපි ගත්ත weight එකත් එක්ක අපි ගන්න අපිට එන average reading purate readings වෙනස් වෙනවා. එතකොට අපිට කොයිලාකවත් ගන්න බෑ මේ වගේ average එකක්. Definitely we have to disregard the first reading because it is deviate than others. Okay, you have to disregard the first reading and you have to consider about the other 3 readings. Then you cannot take an average reading here because the weights are different. Okay, the sample weights are different. So since we disregard the first reading, we don't consider about the first weight. And the second weight is 5.0010 gram, 5.0005 is the third and the 5.000 gram is for the fourth titration. So we cannot take average reading here. We read in here and we have to do the calculation for all the 3 readings. That is the proper way. Okay. මට ඔයගලන්ට කියන්න පුළුවන්කම තිබ්බා ඒක තමයි ලේසි විදිය ගොඩක් කට්ටිය කරන්නේ නැත් ඒක නමුත් ඒක නෙමෙයි ඇත්ත විදිය. නමුත් හරි විදිය. හරියට කෙමිස්ට්‍රි ඉගෙන ගන්නවා නම් ඒ විදිහට ඉගෙන ගන්න. හැමතිස්සෙම ඇවරේජ් ගන්න බෑ. පැහැදිලිද? අපිට මේ වගේ තැනකදී අපි හරියට කෙමිකල් නැත්තම් කීමෝමෙට්‍රික්ස් අපිට වෙනම සයන්ස් එකක් තියෙනවා කීමෝමෙට්‍රික්ස් කියලා. කීමෝමෙට්‍රික්ස් ඇප්ලයි කරනවා නම් ඔයගොල්ලෝ හරියට දැනගන්න ඕනේ. ඇප්ලයි කරනවා නම් මේ readings වලට අපි වෙන වෙනම calculation කරලා ඊට පස්සේ final එකේ average එක තමයි ගන්න. Okay, so we have to do the calculate the acid value for separately for these three readings, and at the end we can take the average acid value. So that's the proper practice. Okay, so average endpoint of the blank titration, we can get that 0.05, 0.05, 0.05, and then what is the average endpoint of the blank titration? So calculate this with me. Then what is the average endpoint of the blank titration for the blank titration? Okay. Then for the second titration, we can disregard the, or we can neglect the first titration because it is deviated than other three, and we have to consider the second titration onwards. For the second titration, endpoint of the second titration is 10.25 milliliters, and the amount of potassium hydroxide required to neutralize the second oil sample is, we have to reduce 0.05 from that burette reading, because 0.05 was used to neutralize the reagents. Okay, we need only the amount of KOH required to neutralize the sample, not the reagents. Okay, so that's the use of doing a blank titration. So you have, so by reducing that, we can take it as 10.20 milliliter. Okay, make a පැහැදිලිද?
Excuse me sir, average value එකක් ගන්නද එතකොට?
නෑ, average value එකක් ගන්න. average value එකක් ගන්නවා නම් වැරදි. මොකද අපි ගන්න weights වෙනස්නේ. අපි දැන් පළවෙනි sample කියන්නේ අපි ගන්න sample oil sample එකේ weight එක වෙනස් වෙන්න පුළුවන්. ඒතර average value එකක් ගන්න එක වැරදි සම්පූර්ණ. ඔයගොල්ලෝ අන්තිමට ඒකේ තේරුම් ගන්න. හරි. එතකොට මේක පැහැදිලි වෙන්නේ මේ blank titration නම් ගත්තේ ඇයි blank titration reading එක අඩු කරේ ඇයි කියන්නේ. හොඳයි. ඊට පස්සේ so amount of KOH moles required to neutralize the second oil sample, you can calculate that we have used 0.1 in KOH, so you can calculate by this. So it's about 1.020 into the power 10 to the minus 3 mole. So weight of KOH in grams required to neutralize the second oil sample. So how can you calculate that? We know the molecular weight of KOH. So what is the answer for this? Weight of potassium hydroxide in grams required to neutralize the second oil sample. So you can do whatever but at the practical exam, if you don't catch this clearly, you will be in a trouble. So what's the answer for this? So 57.12 into the 10 to the power minus 3. That's fine. Okay, so that's your answer. Okay, then weight of KOH in milligrams required to neutralize the second oil sample. We have to convert that into milligram, because if you can remember, in the definition we have to consider milligram of KOH required to utilize 1 gram of oil. So we have to take amount of KOH in milligrams. Okay, so it's you have to multiply it by 1000 to convert grams into milligrams. You know that, you know these things, these are very basics. So it's 57.12 milligram. Okay, පැහැදිලි නේද? gram milligram කරන්න 1000න් වැඩි කරන්න ඕන. gram එහෙම නේද? milligram gram කරන්න 1000න් බෙදන්න ඕනේ එහෙමනේ. පැහැදිලි නේද? මාළු දේවල් නෙමෙයි.
Yes sir.
Okay. Then therefore amount of KOH in milligram consumed to neutralize 5.001 gram 5.0010 gram of the coconut oil sample. පැහැදිලිද? මෙතන තමයි ගැටලුව තියෙන්නේ. අපි average value එකක් ගත්තා නම් මේ weights වෙනස් වෙනවා. මෙතන මේ weights වෙනස් වෙනකොට අපිට මෙතන weight එකේ average එකක් ගන්න බෑ. එතන අපි දැන් 5.0010 අපි මේක 5.50 වුණා නම් ඒ දෙකේ weight average එක ගන්න එක සම්පූර්ණයෙන් වැරදි නේද? මෙතන මේ volume එක consume වුණේ මේ weight එකට. එතකොට අපි ඒක separately හිතන්න ඕනේ. Okay, so therefore amount of KOH in milligram consumed to neutralize the 5.0010 gram of the coconut oil sample, it's 57.12 milligram. So hence amount of KOH in milligram consumed to neutralize 1 gram of oil of the given coconut oil sample. May 57.17 milligram consume mune 5.0010 gram. According to the definition of acid value is defined as the amount of KOH in milligrams that is required to neutralize 1 gram of oil. Okay, so in order to amount of KOH required to neutralize 1 gram of oil, we have to divide it by 5. Okay, this should be 57.12. I did a mistake here. I have done a mistake here. Okay, 57.12. So what is the answer for this? Is this answer correct? 11.42 milligram KOH per 1 gram coconut oil. Is this answer correct or wrong? Please tell me.
Correct.
Then therefore acid value of the given coconut oil sample is this. Acid value එක කියලා කියන්නේ මෙන්න මේක. පැහැදිලිද? එතකොට දෙවෙනි sample එකට acid value එක කියන්නේ මෙන්න මේක. දෙවෙනි titration එක ඇසුරේ. Okay. Acid value is defined as the milligram of KOH required to neutralize 1 gram of oil. Okay, so the units of acid value is milligram of KOH per 1 gram of oil.
Excuse me, sir.
Yes.
Sir, අපි දැන් type මේ second sample එකට විතරද මේ calculation කරන්න ඕනේ?
නෑ. ඔයගොල්ලෝ third sample එකකට කරන්න ඕනේ, fourth sample එකකට කරන්න ඕනේ. sample A ඔක්කොටම කරලා ඊට පස්සේ average acid value එක තමයි අපි consider කරන්නේ. So you have to do this same calculation for the third titration and fourth titration. You have to calculate the acid value, and for that particular coconut oil sample to get an accurate acid value, you have to take the average. එතකොට ඔයගොල්ලන් තව හිතෙන්න පුළුවන් මේක ඇයි මෙහෙම average එක ගත්තේ නැත්තේ කියලා. මේ procedure එකේ තියෙන විදිහට අපිට ගන්න ඕනේ 5 to 6 grams coconut oil. හරිද? එතකොට හිතන්න දැන් මෙතන අපි 5.0.1.1 ගත්තා. දෙවෙනි එක විදිහට අපි 5.5ක් ගත්තා. හරිද? තුන්වෙනි එක විදිහට 5.8ක් ගත්තා. 5.7 අතරනේ. එතකොට අපි අර average value එක ගන්න එක සාධාරණද? මේ burette reading එක. සම්පූර්ණයෙන්ම වැරදි. සම්පූර්ණයෙන්ම වැරදි. මොකද මේ 5.0.1.1 තමයි මේ 10.25 ආවේ. එතන මේක 5.8ක් වුණොත් එම මේ 10.25 නේ මීටර ලොකු වැලියු එකක් වෙන්න පුළුවන්. Okay, so if we take that 5.90, that readings also may be different. Okay, so since we got all the weights near to 5, so there cannot be this kind of higher deviation. So that's why we disregard this. Okay, so if the first reading is about 1 point or 2 point like weight, so this can be this kind of reading, 6.35. Okay, but all the readings are similar to 5. So burette readings, all the weights are similar to 5. Burette readings also in a close precision. Okay, the readings should be in close together. Okay, here you can see acid values, actual acid values for different oils. So for good condition coconut oil, the acid value should be in between 1.99 to 12.8. So if acid value is higher than that, that means that oil is not good for our health. So good for the use. Okay, so acid value is different from oil to oil, depending on the triglycerides and the ratios of triglyceride present in particular oils. Okay. Oh. First and fourth readings වෙනස් වෙන්නේ ඇයි කියලා අහලා තියෙනවා. හරි. ඒක තමයි. ඒක හොඳ ප්‍රශ්නයක්. ගොඩක් හොඳ ප්‍රශ්නයක්. දැන් වස්සලා අහලා තියෙනවා first and fourth readings වෙනස් වෙන්නේ ඇයි? ඒ වගේ weight සමානයිනේ. ඒක තමයි. first and fourth readings වෙනස් වෙන්නේ ඇයි? ඒකේ weight සමානයි. ඉතින් මේක අපිට වැරදීමක් වෙන්න ඇති මෙතන. ඒකයි අපි disregard කරේ. අපි titration එක කරද්දී අපි වැරදියට end point එක read කරලා වෙන්න ඇති. එහෙම උනේ නැත්තම් මෙන්න මේ reading එකයි මේ reading එක සමාන වෙන්න ඕනේ. එතකොට then we have to disregard the unit readings or the whatever the measurements when we did something wrong. Okay, so there can be errors in every chemical experiments. So the error is not in acceptable level. We have to disregard the measurements or the readings. So that's what happened here. Maybe we have underestimated the endpoint. So I think that's the answer for your question. Is that clear, Vatsala?
What? Thank you, sir.
Badly.
Yes,
Ekay
sir.
api meka disregard karana. Eතන මේක සම්පූර්ණයෙන්ම වැරදි reading. Okay. So
Yes, sir. Thank you, sir.
okay. So that's the reason for not considering that. So do you have any questions regarding this? So you have to submit a practical report for this experiment, acid value, determination of acid value. Okay. So even you have to understand why we didn't get the average titration readings. Okay. Sometimes we cannot do that. First reading ගන්නේ නැහැ නේද කියලා අපිට first reading if it is in the good usually in chemical experiments we do the first reading to get an approximate endpoint. So okay, so to get an approximate endpoint but it is very close to the other reading. So when we don't have facility to conduct four titrations, that's okay to take the first three when there's no much deviations. හරි එතකොට තව දෙයක් මම පුංචි දෙයක් කියන්නම් පුංචි දෙයක් විනාඩි දෙකකින් අපිට KOH lab එකේ නැත්තම් අපි මේ වගේ දේවල් තමයි දැන ඉගෙන ගන්න ඕනේ. අපි conditions handle කරන්න. KOH lab එකේ නැත්තම් sodium hydroxide විතරක් නේ lab එකේ තියෙන්නේ. අපිට මේ acid value එක හොයන්න පුළුවන්ද බැරිද? මොකද ඔයගොල්ලෝ හිතන්නේ? So according to the definition, acid value is defined as the milligram of KOH that is required to neutralize 1 gram of oil. So what's your answer, sir? Someone has answered to me. පුළුවන්. Definitely පුළුවන්. අපි sodium hydroxide වලින් මේ titration එක කරනවා. Sodium hydroxide වල titration එක කරපස්සේ අපිට moles sodium hydroxide මේකේ moles ප්‍රමාණය හොයාගන්න පුළුවන්. Amount of sodium hydroxide mole required to neutralize the second the oil sample. Okay, that mole is equivalent to KOH. පැහැදිලිද? sodium hydroxide moles මෙච්චර ගියා නම් මේ oil එකේ free fatty acid neutralize කරන්න ඒ ප්‍රමාණයම තමයි sodium potassium hydroxide. නමුත් potassium hydroxide weight එක වෙනස් වෙනවා. මොකද molecular weight එක වෙනස් වෙනවා. නමුත් moles ප්‍රමාණය සමානයි. Okay, so even you have provided with sodium hydroxide, you should be able to do this titration. You have to be vigilant. Okay, sometimes we have to do, especially countries like Sri Lanka, we have to work on restrictions. So no point of saying that we don't have enough facilities, but we have to be vigilant and work on whatever the resources we have. Okay, if you are talented enough, you can think and do and get the maximum out of the resources we have. පැහැදිලිද? ඒක තමයි එක්ස්පෙරිමන්ට් වලින් තියෙන වැදගත්කම. ගන්න හිතන්න පුරුදු වෙනවා time manage කරන්න. ඒ වගේම දැන් අපි ලස්සන demonstration slide එක ඔයගොල්ලන්ට lab එකේ practicals කරන්න පුළුවන්. ඉතින් chance එකක් තිබ්බා නම් ඒගොල්ලොත් ඒක වැඩ කරන්න තිබ්බා. ගොඩක් දේවල්. ඒ වගේම අපේ day to day life එකට වැදගත් වෙන දේවල් තියෙනවා. Biochemistry is very important subject. One of the very important subject in your curriculum. Okay. Okay, then if you have any questions, feel free to ask. You can contact me. You have my email address as well as my phone number. Yes. First reading it calculate කරන්නේ නැද්ද? ඒ reading it වැරදි නිසා. In here, we don't consider the first reading because it is not in good match with other readings. Okay. But if it is in the good match with other readings, it's okay to use that. Okay. So if you don't have any questions, you have to submit a practical record for this, and I will upload the particulars about that to your LMS, and you have to do the calculation. So you have to determine the acid values of the two samples and compare them. Okay, and you will understand the acid values of old coconut oil samples as well as a new coconut oil sample. Okay, so with that I will wind up the session, and we will meet in next Tuesday. Okay, so thank you for your understanding, and I think you all got the content clearly. Thank you.